Hi there, this is Liz, and in the next few minutes we're going to have a look at some enzymes. So enzymes are proteins that catalyze reactions. The chemical reactants that enzymes bind to are called substrates. So enzymes are biological catalysts. So these are substances that speed up a chemical reaction without being used up in the process itself. Enzymes are proteins that catalyze biochemical reactions, so inside the body, inside cells, or inside some sort of substance in the body. Enzymes can act inside or outside of the cells. For a chemical reaction to start, it needs a specific amount of energy, and this amount of energy is different for different reactions, and we call this the activation energy. Enzymes lower the activation energies of chemical reactions inside cells to increase the rate of reactions. Enzymes lower the activation energy by binding to the reactant molecules and allowing chemical bond breaking and bond forming processes to happen more easily. Enzymes catalyze specific reactions. The active site has a specific shape for each enzyme. Substrates with a complementary shape to the active site of an enzyme can bind to form an enzyme-substrate complex. The shape of the active site is determined by the tertiary structure of the protein. So, the active site is determined by the tertiary structure, and substrates can bind to the complementary active site to form an enzyme-substrate complex. So how do enzymes work? We call a model of enzyme action something which describes how enzymes work. And over time, there have been lots of different ideas about what this model is. So how do enzymes work? For many years, it was thought that enzymes worked in a lock and key manner. But now there's a new model called the induced fit model, which is actually more widely accepted. So let's first go over the lock and key model. The lock and key model was originally used to explain enzyme action. The model proposed that enzyme and substrate fit together perfectly. For example, the substrate is a key fitting into a lock, which is the enzyme. So imagine when you put your key into a lock, it fits in perfectly because the lock has been made for the key, or the key has been made for the lock. And when you turn that, you open the door. I guess when the substrate fits into the enzyme, you're allowing a reaction to happen. The other model is the induced fit model. This suggests there's a more dynamic interaction between the enzyme and the substrate. The model states that as an enzyme and substrate come together, the interaction causes a small shift in the enzyme structure. The shift means that the enzyme and substrate can bind to form an enzyme-substrate complex and catalyze a reaction. And this model is more widely accepted now. So, what is an enzyme-substrate complex? Enzymes bind with substrates to form an enzyme-substrate complex in a specific fashion. The specificity of an enzyme is determined by a few things. One of these is the active site. Every enzyme only catalyzes one specific reaction, and every enzyme has a specific active site that is complementary to the specific substance. This jigsaw puzzle-like match between an enzyme and the substrate is what makes enzymes highly specific. Environmental factors also determine the specificity of enzymes. The 3D tertiary structure of the polypeptide chain determines the shape of the active site. Environmental changes can change the tertiary structure of the active site and stop the enzyme from working properly. This would be called a denatured enzyme. For example, if the temperature is really hot, this can cause bonds to break, and this will then affect enzyme structure. And pH changes can also have a similar effect. And if all the bonds aren't there, then their structure is different, and that would change the shape of the active site. So the substrate can no longer fit properly into that active site. And that pretty much sums up everything you need to know about enzymes. So in summary, enzymes speed up chemical reactions, enzymes are proteins, and enzymes bind with substrates to form an enzyme-substrate complex. And how they bind together is best explained by the induced fit model.